Let's start off by understanding what homogenization is. Using the example of milk, homogenization is the process that allows fat droplets to disperse throughout the milk, so the milk stays consistent and won't separate into layers. Here are some key vocabulary words that you need to know for this lab. Hydrophobic molecules are molecules that stay away from polar or charged molecules such as water. Common examples of hydrophobic molecules are oil and fat. Hydrophilic molecules are molecules that stick or mix with charged molecules such as water. Common examples of hydrophilic molecules are salts. Molecules are particles that are made up of a group of chemically bonded atoms. Atoms are the tiny building blocks of all matter. And finally, density is the amount of mass per unit of volume and is how tightly packed and heavy matter is for its size.
Molecules in a solution tend to aggregate or group together with other molecules that are similarly charged. For example, fat molecules will clump together with other fat molecules, while water molecules will want to stick with other water molecules. Milk is made up of different molecules such as fat, water, and protein. In order for all of those different types of molecules to stay together, the milk has to go through a process called homogenization, or else they start to separate into different layers. However, even after milk goes through homogenization, some fat molecules continue to stick together. We will be able to see this in action in real life, and so it's experiment time.
Step 1. Pour milk into the bowl. You don't need too much. Just make sure it's enough to cover the bottom of the bowl, just like we have right here. Step 2. Wait for the milk to stay completely still for a few seconds. While waiting, predict and write down or draw what you think will happen. Step 3. Add 1 drop of food coloring to the milk. Do not mix or stir. Step 4. Take a cotton swab and dip it into the dish soap. Step 5. Use that same cotton swab and dip it into the milk. Slowly move it towards the drop of food coloring. Step 6. Continue to move the cotton swab around and observe how the coloring is moving. Step 7. Try adding more food coloring or different colors to observe what happens differently. You are now done.
Now that you've done the lab, what did you notice? What happened when you tried to move the cotton swab closer to the food coloring? Did the color stay in one single shape, or did its shape completely change? Did the milk react at all? What changes did you notice in the food coloring? Was your prediction correct or completely different? Take a minute to think about what happened and try to answer these questions.
Now you might be wondering what's going on. Well, don't worry, there's an explanation. Have you ever tried mixing oil and water together? The fat molecules in oil, just like those in milk, are hydrophobic, meaning they don't like charged molecules such as water. These hydrophobic molecules will do their best to stay away from water, and in order to stay away, they clump together. When all of the fat molecules clump together, they start to float. This is because their density is lower than water, allowing them to float up to the surface like a pool noodle, preventing the oil from mixing with water. Dish soap is a detergent. This means its molecules have a hydrophobic end and a hydrophilic end too. Hydrophilic molecules like sticking with water molecules. Because the dish soap has molecules that like both fat and water molecules, they're able to form a bridge between them. This causes the clusters of fat molecules at the top to break apart and disperse in order to stay away from the water.
In milk, the fat molecules at the surface break apart into smaller clusters and are pushed away from the dish soap. When we see the color spread out, we're actually watching the fat molecules spread out.